Now, cleavage means division of the cells. Now, whole embryo is formed by the start of cleavage. Cleavage means division of the cells. Now, the first thing which occurs after ovulation, when the oocyte is fertilized and becomes zygote, it is at the moment is a single cell. This single cell go on dividing and dividing and dividing to form the whole of the embryo. So every organ of the embryo, like liver, heart, brain, everything, that is formed by the mitosis phenomena. The meiosis occur only in gonads, testes and ovaries. The mitosis which occur and start from the this zygote, and that is the induction of the cleavage which occur after the zygote is formed. Cleavage occur or division. Cleavage means division or mitosis phenomenon that occur 30 hours after the ovulation. That means almost 1 day and these 6 hours after the ovulation, zygote undergo series of mitosis, repeated mitosis, in order to increase the number of cells. So there is increase in number of cells. Different organs are differentiated, like eye is developing, brain is developing, liver developing, pancreas developing, uterus, bladder, kidney, like this. Their size increase, or they are initially in the form of small bud. Later on, it acquires the full structure, like full kidney is formed. So that is by the phenomena of the mitosis. And mitosis, so that increase the number of cells. Smaller cells called blastomeres. Cleavage or zygote that divide into the smaller cells, that is in the form of the dichotomous division, that is 1 divide into 2, 2 divide into 4, like this. So the cell which are initially formed, like 4 cell stage, zygote change into 2 cell stage, then 2 become 4, 4 become 8, 8 become 16, and similarly 16 become 64. So they are called the blastomeres, and they form the loosely arranged clump. So these collection of cells initially, the ball of cells you can say, which is formed say up to 64 cells, their name is given at the moment blastomeres. Blastomeres are small cells that is derived from the single zygote, and they form loosely arranged clump, which means the cells are initially they are loosely arranged in the form of a group or clump. Blastomere, because this phenomena is the cell is oocyte is surrounded by zona pellucida, which is the tight layer around the that is the oocyte. Around that is the zona pellucida. Those division of the cell occur inside zona pellucida. The blastomere, because there is not much space inside the zona pellucida, so cells division which is continue, it is impossible for the cells to uh, accommodate themselves in zona pellucida in the loose form. As we said, they form the loosely arranged clump. They can no more be loosely clumped inside the zona pellucida because the number of cells are increasing. So what happens is a blastomere, in order to accommodate inside the zona pellucida, they maximize their contact with each other. That is, maximum contact occur with each other. How the maximum contact to accommodate themselves inside the zona pellucida is they become flat. Instead of rounded cell, they become flat. Their one surface become flat and they are attached by tight junctions which is made up of glycoprotein. Tight junction is present and cells in order to become accommodate in less space inside zona pellucida and more cell that can be accommodated by flattening, no more rounded. So these are their surface become flat. They they are flat and have a tight junction between them, that is the glycoprotein junction. So they form a compact, which means solid almost, ball of cells held together by tight junction, process called compaction. Initially, what we said over here that they are loosely arranged because the number of cell is less, space is more. As soon as the number of cell is increasing, so they become flattened and tightly packed with each other, and the process is called compaction, which means they become a compact structure. In compaction, cells flatten on each other to maximize intercellular contact with tight junction. 12 to 32 cells blastomere are called medulla. Normally, what is the name is given 16 cell stage is medulla, or between the 12 to 32 cells blastomeres when they are formed by the dichotomous division 1 into 2, 2 into 4. That is the that blastomere that is called medulla. That mulberry shape. Medulla forms 3 days after the fertilization. That is the once the fertilization occurs, then within 3 days after fertilization, the medulla is formed, which means that the oocyte which enter in the fallopian tube after ovulation 14 days पे and enter it and it is get fertilized say within 24 hours, then in next 3 days that is remain inside the fallopian tube and undergo division and form the medulla. That is the 12 to 32 cell stage is achieved inside fallopian tube. The medulla formed 3 days after fertilization, which means it spent 3 days in fallopian tube and change into the medulla or 16 cell stage or 12 to 32 cell stage. These cells now organize as inner cell mass, which is the embryoblast, which will develop into embryo. Now, these cells, which is previously the ball of cells, now change into or organize itself into two groups, that is the inner cell mass and outer cell mass. Inner cell mass cells are less as compared to outer cell mass. Inner cell mass division cells that form the embryo, which will develop into embryo. And outer flat cells arrange on periphery as outer cell mass develop into trophoblast or placenta. So there are the two things which are present important in the development of the human fetus. That is, one is the embryo or fetus, which is formed, embryo is formed. Second thing which is important to develop is placenta for nutrition, excretion, and the oxygen supply to the fetus. And that is very important part for development of the embryo. So outer cell mass develop into trophoblast or placenta. A space is created in between these is blastocystic cavity. Now it absorbs the fluid and that by reorganizing cells. They are present on one side, inner cell mass. On periphery, their organized cell is the outer cell mass. In between them, a space is created, and that space is name is given blastocystic cavity. It is now divisible into polar and apolar ratio 8 into 8. The outer polar, so this is the what's shown two cell stage. Zygote which was single now divide become this. This is covered over by zona pellucida. Now two become four. That this one divide into this two cell. This one divide into these two cell. It become four. Similarly, these are the 16 cell stage medulla, and that is achieved in 3 days. Medulla. Blastomeres that is surrounded by zona pellucida. This is one. Zona pellucida. This is the initial picture which is occurring. That two cell, four cell, then go on. Now, these cells which previously you have seen that is forming the this compact ball of structure now organize itself into two groups. This is the one group of cell. There are less number of cells, 7 or 8, says, and rest of the cell that organize on the periphery like this. And this is also on peripheral cells. This all is peripheral arrangement of the cells, and this is the inner cell mass. This will develop into embryo, and outer cell mass which will form the placenta or trophoblast. Name is given trophoblast. By this organization, that one group is on one side of this cyst and the other is on periphery organized, and in between them here is present blastocyst cavity. That is the so cavity is automatically that is created. This is the peripheral arrangement, outer cell mass. This one is the inner cell mass. Now the outer cell mass, another name is given trophoblast. That is the we divide into two groups. Outer cell mass on the embryoblast, covering the embryoblast. This one, this is the this part which is covering the this inner cell mass. This is the inner cell mass. It is covered on outside by these trophoblastic cells. This component of the trophoblast name is given the polar trophoblast. Polar. Toward the this is a we actually divide the cyst into two components. Why the name is given cyst? Because a cavity is created. Cyst is the cavitated ball-like structure. That name is cyst, and it absorbs the fluid from outside, which having nutrition, and collected in this chamber. It is not empty. It is filled with secretion, which is absorbed by trophoblastic cells from the fallopian tube or from the uterus. Uterine secretion, which are uterine glands are producing, that is absorbed by. This is act as a permeable membrane like this, and selection occur for absorption of the different that is the nutritive fluid. It is accommodated over here. So name is given blastocystic cavity. Now this is the we divide the cyst into two components. Inner cell mass, this component is polar trophoblast, and this rest is which is not covering the inner cell mass. Its name is given mural trophoblast. Mural mean cavity. MURL. Mural mean cavity. So that is surrounding this cavity. This part will start from here and up to this level. This is the mural, and this component which is seen is the polar trophoblast covering the inner cell mass. This is the blastocyst cavity. Here is shown that on now blastocyst, when it is formed inside the fallopian tube up to third day after the ovulation. That is, this structure is formed. Morula is changed into blastocyst. It enter into the uterine cavity from the uterine ostium of the fallopian tube, enter in the uterine cavity, where on the fourth day it enter in uterine cavity on fourth day, where it remain for next 2 days, 4th and 5th day, and remain floating in the uterine cavity and absorb the secretion. This layer act as a selective membrane, absorb the secretion which is accommodated in this cavity, and it collect the nutrition, you can say, for the next 2 days, and 4th and 5th day is spent in the uterine cavity. On 6th day, it start attaching to the endometrium, which was prepared by the progesterone, and that is the secretory phase of the, that is start from the uh, 14 days to 26 days that of the menstrual cycle. This bed was prepared under the progesterone, and there is attachment of the blastocyst. In this manner, it is attached and try to penetrate inside the endometrium. This cyst will see next that cyst will enter into the endometrium and disappear from the uterine cavity. Now, blastocyst around it, what we said, that is surrounded by the zona pellucida, eh? And that is the surrounded by zona pellucida. Zona pellucida breakdown, and there is no more zona pellucida around the blastocyst is seen. That breakdown, and this blastocyst hatches out or come out from the zona pellucida, uh, primarily from the zona pellucida. It hatches out from a small hole in the zona pellucida, and it develop a figure of 8 like this. This is the figure of 8, and that is the this figure of 8. It hatches out from the, and zona pellucida disappear. Now it start attaching, and not only attach with the endometrium, but it rather penetrate into the endometrium, and then further growth occur in the inside the endometrium. Cell contribute largely to trophoblast. Outer cell contribute large, but polar cells contribute inner cell mass. Trophoblasts engage in fluid transport, as I told you, act as a permeable membrane. Fluid transport to generate a cavity that expands at 32 to 64 cell stage and convert the medulla to sphere, a rounded ball-like structure sphere. The blastocyst, once sphere, the name is given blastocyst. That is, the cyst cavity is created. Once blastocyst form, no further division of the cell occur. No further division. Formation of the blastocyst. Blastocyst hatches out from the zona pellucida at 6 to 7, yeah, Patches term derived from just like a chick embryo, which break down the eggshell and it come out from a slit or hole in the eggshell and come out from it. That term used hatches. From the zona pellucida 6 to 7 day, assisted by enzyme like trypsin. That is on zona pellucida. This is the zona pellucida, and a small hole here is created by the trypsin enzyme and blastocysts that come out in a figure of 8. You can see this picture. That is by punching it come out slowly. Half when it is half is out, half is in. It gives a picture of 8 figure. And inner cell mass is present over here, whereas the outer is like this. So sometime when during hatching, it break down at this point. So inner cell mass half is remain here, half is in here, and they become twins. They become twins. That is blastocyst divided into two components. At the what we say division of the blastocyst at the inner cell mass. But normally it doesn't break. It totally completely come out. It oozes out of the small slit as figure of 8 bisected by zona by pellucida. Outer cells are flat which possesses features of transporting epithelium. These outer cells are flat, trophoblast, and that have features of transporting epithelium to collect the fluid inside the cavity, derived from the uterus. Inner cell mass is arranged as upper layer closest to the polar trophoblast. This side is known, name is given polar trophoblast. Toward that, where the inner cell mass is present. A one side inner cell mass is present, and that side trophoblast name is given polar. Rest of the trophoblast is mural. Mural means cavity, which is lining the cavity. That which is covering the inner cell mass is polar trophoblast. Trophoblast. Inner cell mass now name is given here the epiblast, single layer. It is a single layer structure. Now, outer cell mass is also a single layer structure. Inner cell mass, which is the name given epiblast, is a single layer structure and a single cavity is present. So, this is the end of first week of the development. What we say. First week mean the single layer is formed, which mean yes, trophoblast is single layer and inner cell mass is single layer. That is up to first week is single layer. In second week, when we enter, the inner cell mass will change into double layer structure. Outer cell mass will also change into double layer structure. That will be the second week. And give embryonic cells and a lower layer hypoblast. That this epiblast will divide into another layer below it. Here we will show you next. That is the name is given hypoblast below the epiblast. For extraembryonic fate, on sixth day, blastocyst adhere to the uterine mucosa. I told you that fourth day it enters in the uterine cavity. Fourth and fifth day that is spent in the or floating in the uterine cavity, absorbing the secretion derived from the uterine glands, and it accommodates more and more nutrition. Then on sixth day, blastocysts adhere to the uterine mucosa. Steps are number one, dissociation of zona pellucida. That is zona pellucida is that is the dissolve. Second is the adhesion of the blastocyst to endometrium of the uterus. That first it attach adhere. Third step is penetration into endometrium and migration of the blastocyst. That is the migration to the or entry into the endometrium. Then number four is spread and proliferation of the endometrium. That is endometrium further proliferate and spread out. These are the four steps. Merula enter into uterine cavity on fourth day after ovulation. Next 2 days remain floated in the uterine cavity up to 6 days and take secretion from the uterus. On 6th day, it attaches the uterus. Normal site of implantation. What is the normal site? This is important. Where the normally that is the blastocyst implant, and that is on the posterior wall near fundus in median plane. Here as is shown, this is the anterior wall of the uterus. This is the posterior wall of the uterus. Upper part is fundus. So it occupies the posterior wall and fundus, where the maximum proliferation of the endometrium is there, or the proliferative phase, or the secretory phases of the that is the third phase under the progesterone. This bed is prepared as described in the menstrual cycle, that is progestational phase, where the this bed is formed under the progesterone, 14 days to 26 days. Now, where this bed is maximum formed, posterior wall and fundus, and here the endometrium, that is blastocyst will embed normally. This is the normal. Medulla enter into uterine cavity. Fluid began to penetrate through zona pellucida, inner intercellular spaces of the inner cell mass, spaces coalesce, that is spaces combine to form the blastocoele, embryo guard blastocyst. Zona pellucida disappear. Trophoblastic cell at embryonic pole begin to penetrate between the epithelial cells of uterine mucosa. So trophoblastic cells which are toward the embryonic pole, that is the site where it start entering into the endometrium. Trophoblastic cell at embryonic pole begin to penetrate between the epithelial uterine mucosa. On 6th day, L-selectin on trophoblast cells. On trophoblast cells, there is L-selectin. Here is shown that is 2 cell stage, 4 cell stage, 8 cell stage, that is, and this is the 16 cell stage or medulla. Initially, it is a compact ball of cells surrounded by zona pellucida. Then these cells are organized in such a way that one is toward the one side, inner cell mass, it will develop into embryo, and other is this peripheral cells all around is the trophoblast. Now cells which are covering this inner cell mass, this one, this component, that is the polar trophoblast, and rest is the neural trophoblast. Here is the again shown blastocyst cavity. These are the peripheral range cells, trophoblast. This is the inner cell mass or embryoblast. Of blastocyst, L-selectin of blastocyst. There is a L-selectin on the blastocyst. Here is shown. Now the polar trophoblast site is the site where it penetrates. Now it has L-selectin, that is receptors, and that on the endometrium there is also present the receptors or binding protein which bind with this L-selectin. L-selectin on trophoblast, carbohydrate receptors on endometrium of the uterus or the endometrium of the uterus. that is the on which is present the carbohydrate binding protein and carbohydrate receptors which receive this L-selectin. L-selectin which is present on the toward the polar trophoblast, toward that part which is surrounding the that is embryoblast. Now they will make the contact with each other. In between them, what is said that some leukocyte cells derived from the mother blood vessel, the uterine blood vessels, that also play the role to binding them in between a leukocyte cells, white cells. Carbohydrate receptor on uterine epithelium, L-selectin of the blastocyst, and carbohydrate binding protein and carbohydrate receptors are present on the uterine epithelium, mediate attachment of blastocyst to the uterus. Further invasion, once it is attached by this mechanism, that is L-selectin here and carbohydrate receptors on the endometrium. Once it is attached, then two things come in play. Further invasion by trophoblasts, that is by the integrins. What is integrin? Expressed by trophoblasts, that is present on this is integrin which is present on trophoblast and expressed by trophoblast and extracellular matrix laminin and fibronectin. This is the laminin receptors, this is the fibronectin which are present that come in action. Integrin receptors for laminin that is promote attachment. So role of the integrin that is the laminin is attachment of the blastocyst to the endometrium, whereas the fibronectin, laminectin and fibronectin, integrin receptors for laminin. What is the role of laminin? It promote attachment, while the fibronectin stimulate migration. That fibronectin control the migration or penetration into the endometrium. So it give attachment, it help in penetration. Promote attachment, while the fibronectin stimulate migration. Interaction involves in between them. I told you that leukocytes and endothelium taken from the flowing blood into the uterine wall. Implantation of blastocyst, which means penetration into the endometrium. Endometrium, actually blastocysts disappear from the uterine cavity and totally embedded in the endometrium. The term used is implantation of the blastocyst. It implant or totally disappear from the uterine cavity and embed in the bed prepared in the secretory phase of the uterine, that is the menstrual cycle. What is the this bed is under progesterone, which has tortuous blood vessels, tortuous glands, and ready for reception of the this coming guest. It is completed in second week, 6 to 10 days after ovulation. Trophoblasts differentiate into two layers. Cytotrophoblasts show active mitosis. The previously cytotrophoblast, which was the outer cell mass or single layer, it shows active mitosis and develop this red layer around it. And this is the syncytiotrophoblast. Now the name is given. And the primary layer trophoblast name is given cytotrophoblast. So initially the name which was given the outer cell mass, now name is given cytotrophoblast. These are well organized cells, have the regular wall of the cell, whereas the syncytium is they lose their cell wall, cell membrane. So in cytotrophoblast, cell membrane is well organized. In syncytio, cell membrane that disappear and it become a mass of protoplasm having multiple nuclei in it. Cytotophoblasts show active mitosis. Cell migrate to form sensitive tophoblast layer where they fuse and lose their cell membrane. They fuse and they form the loose cell membrane. Now we have entered into the second week of the development, which is the eighth day or ninth day. Now everything in second week will become double. Outer cell mass will, which was previously a single layer, now become double layer. Outer sensitio and the first primary layer name is given cyto. Similarly, inner cell mass will also divide into epiblast and hypoblast double layer. Here is shown the various characteristics, and that is the cytotophoblast layer. You can see over here. Their cell membrane is well organized. You can see here. That is, you can recognize the cell membrane. Regular cells are present. These are the regular cells which are bounded by the cell membrane. Now, it divides by mitosis and develop a new layer around it. This new layer, and that is the sensitive tophoblast. But here you can note one point is that no cell membrane is present in sensitive tophoblast. When no cell membrane is present, this is just a mass of protoplasm and having the nuclei in it. That is the it is the nucleated mass of protoplasm, not the regular cell like this cytotrophoblast. Previously, it was only trophoblast. Now, when it develops the second layer, name is given, primary layer name is given cytotrophoblast, and outer is the sensitive trophoblast. As it embeds inside the endometrium, this totally inside endometrium, here is the uterine cavity from which it has disappeared. The defect, this is stratum compactum, this one, and it make a hole in it. This is the site of entry, or this is the site of entry. This one from where blastocysts enter. When it fully enters, this defect is closed by a fibrin tissue, which is the name is given fibrin coagulum. That is coagulate over there, fibrin material, and close this defect. And now it is totally embedded inside the, and this will drive its nutrition from the uterine gland secretion, which are present near to it. That is absorbed by syncytio, then cyto, and ultimately approach to the this cavity, and that will be developing in it. Syncytio trophoblast later on that start developing spaces are undergo degeneration phenomena. By these spaces are created. These spaces are name is given lacunae. This is the trophoblastic lacunae. Lacunae means small spaces which are created in the syncytia trophoblast. And syncytia will make contact with the blood vessels of the uterus, and so that will rupture and fill these spaces, become full of blood. Second thing you should note over here, that is the outer cell mass is divided into cyto and syncytio in second week. In second week, inner cell mass also divide. Number one is this was the inner cell mass. We change into long columnar cells. This one, long columnar cells. Name is given epiblast, and cuboidal cells below it is the hypoblast, as the name is given. So inner cell mass, which was previously single layer, epiblast, now become double layer, epiblast and hypoblast. Similarly, cytotophoblasts, which divide into second layer, the name changes. Trophoblast, this primary layer, now name given cytotrophoblast, and newly created layer, that is the sensitive trophoblast. Now, from this This is the hypoblast. There, from this margins, cell divide and line this cytotrophoblast inside, and name is given exosolomic membrane or Husser membrane. This is the this layer is derived from the hypoblast, and this hypoblast then which line this cavity is derived from hypoblast Husser membrane. When it encloses this chamber, you can see or cavity. Now name is given primary umbilical vesicle or primary yolk sac name is given because no yolk is present. So the new name is given primary umbilical vesicle. Another thing you note over here that this is the epiblast which is previously near to this outer layer or cyto that is near to that, but now it starts separating out, and by this separation it dip down, a cavity is created. Now space is created. Name is given amniotic cavity. Now from the peripheral margins of the epiblast, cell divide, and that is the lining of the cytotoroblast. These cells they form the name is given the amnioblast. Amnioblast is the derived from the this epiblast cells. Now bounding by the amnioblast, below is the epiblast, and this chamber is name is given amniotic cavity. This chamber, which is derived by the hypoblast and Husser membrane, is primary umbilical vesicle. This is the amniotic sac. Now you can note over here the previously inner cell mass was single layer. Now it divisible into two layers. So large columnar cells, that is the epiblast, and cuboidal cells, hypoblast, and two chambers are formed. One this primary umbilical vesicle, other is this amniotic sac. So what alteration occur in second week? Trophoblast change into two layers, cyto and syncytio. Inner cell mass change into two layers, epiblast and hypoblast, and two chambers are formed. That is umbilical, amniotic cavity, and umbilical vesicle. So another name given to second week is bivesical stage, which means two cavities are formed, two vesicles. One is this primary umbilical vesicle, other is the amniotic cavity. So two vesicle or bivesicle are formed. Two layer of inner cell mass, epiblast and hypoblast. Two layers of trophoblast, inner cyto, other outer inner. That is the total alteration which occur in the second week. Syncytial trophoblast is penetrating layer. It penetrates into this blood vessels and break down it. So blood now start entering into these lacunae, maternal blood. We'll see in the next. Cytotrophoblast, a rapidly expanding multinucleated mass with no cell boundaries. No cell boundary. Syncytium invades the endometrium and blastocyst slowly embedded at embryonic wall in the endometrium. It get embedded. Syncytiotrophoblast displaces the endometrial cells. It obviously to make the space, it displaces the endometrial cell at implantation site. Penetrated defect is the site where the defect occurs, stratum compactum. Penetrated defect in the endometrium is closed by later on by a fibrin coagulum, that is the clotting of the fibrin, that is clothing plug, and endometrial cells undergo cell death which facilitate the So surrounding endometrial cells by the penetration of blastocysts that undergo death due to pressure. So that facilitate the invasion, so that allow the entry of the blastocyst into the endometrium, as that assists facilitate invasion into connective tissue cells. Around implantation site, the cell which are around the penetration site accumulate glycogen and lipids that accumulate the glycogen and lipids, which is the nutrition for the blastocyst. Syncytium engulf these cells that break down these cells and engulf it, provide a rich source of embryonic nutrition. Syncytiotrophoblasts synthesize at the moment now start, which is almost 12 days after ovulation. HCG, which is entering into the blood in lacunae, the lacunae में जो maternal blood है, that is by the penetration of syncytium, blood is collected in lacunae, that is maternal blood. Now, syncytium produce human chorionic gonadotropin hormone (HCG), which enter the blood in lacunae to maintain the life of the corpus luteum cavity. That is the corpus luteum, which maintain the life of corpus luteum. Early check of pregnancy is by the level of HCG in the blood. You can check this early pregnancy if there are not, that is by checking the level of the HCG in blood. At the end of second week, positive pregnancy test occurs, almost 12 days or so. Now small spaces appear in blastocystum in the early amniotic cavity. Soon embryoblast. Here you can see that syncytial trophoblast has penetrated into maternal blood vessels, and this maternal blood start flowing in these spaces created in the syncytial trophoblast. Name is given lacunae. Lacunae become filled with maternal blood that is derived from the internal blood vessel. Similarly, maternal blood vessels which are eroded by that is the by the syncytium, blood start flowing over it. Now syncytium here produce HCG which directly enter into maternal blood, and the maternal vein carry back the blood to the uterus, and through maternal vein the HCG approaches to the corpus luteum and maintain its life. So blood level of HCG can be checked. This is the hypoblast, this is the epiblast, this is the amniotic cavity, and this layer is green layer is amnioblast, right from the epiblast. This hypoblast, which give a new layer, which is the inner side, this lining, this is the lining, name is given Hasser membrane, this is the primary umbilical radicle, this is the amniotic cavity. This is the coagulum, which is the close from outside. This is stratum compactum. Separate from blastocyst, epiblast, and form the amniotic amnion to enclose the amniotic cavity. Embryoblast cells flat bilaminar plate of cells. The embryonic disc consists of two layers. Epiblast. Epiblast layer, that is the highly columnar cell related to the amniotic cavity. Hypoblast consists of small cuboidal cells adjacent to the exocoelomic cavity. Epiblast form the floor of the amniotic cavity. Obviously, you can see that this is the epiblast form the floor of the amniotic cavity. Hypoblast form the roof of the primary umbilical radicle or exocoelomic cavity, and continue with the exocoelomic membrane or Russell membrane to form the primary umbilical sac are primary umbilical vehicle. At embryonic pole, vacuole appear, which I told you, in the syncytium. When these vacuoles fuse on the large lacunae, so-called lacunar stage, at embryonic pole is that where the inner cell mass present. Opposite its name is given abembryonic pole. This is the, you can say that this is the blastocyst embryonic pole, and this opposite is this one, abembryonic pole. At embryonic pole, only cytotrophoblast is present, as shown in previous picture, and syncytium get thinner. You can see over here that syncytial layer that is more developed on toward the polar side and on the side, but no syncytium toward the abembryonic pole. This hole is the abembryonic pole where the no syncytium is present, whereas the embryonic pole, this syncytium is present. Syncytium is just a mass of nucleus. This is the amniotic cavity. This is primary umbilical vehicle. This one epiblast, hypoblast. This is cytotrophoblast, and outer is the syncytioblast. It is fully embedded in the uterine cavity and establish the communication with maternal blood vessels, and that become filled with blood. Here is the blastocyst is fully embedded in the. This is the just entering, and it is further entering. Then is the almost is entered, and it is fully inside the endometrium. Okay, okay.